Okay, thank you guys all for coming to the templated editing in eukaryotic systems for gene therapy in health and in agriculture. And we are going to get started with the first speaker, Bing Yang. But before that, my co-convener, Aaron Hummel, is going to come introduce him. So thank you guys all for coming, and we're going to get started.
I'm impressed you were able to repeat that whole title from memory. Nice job. So a couple words about the session first. This is a session that is in part here today. We'll have some wide-ranging perspectives on templated editing in various eukaryotic systems in this session. And in some ways, and you'll see that in some places, those kind of systems are being applied today, right now. And in other ways, it's a little bit aspirational, and it's anticipating a future that I believe is probably 2 to 3 years away, where we'll be much more routinely applying templated editing systems in both plant agriculture, animal agriculture, and human therapy. And we believe that for that reason, this was a timely session. We hope to give you a diverse sampling of topics and perspectives. I think you'll get that. It's definitely not a comprehensive look at the field. So we're going to give you a broad, rapid overview and hopefully pique your interest, and we can go deeper in discussions afterwards. So we'll have 20 to 25-minute talks from 3 different speakers. We'll have, to the extent that we can stay on time, we'll have time for a Q&A for each speaker within that time frame. And then if we stay on time, we'll have 10 minutes for discussion at the end, and we'll get all 3 speakers up here, and we can do Q&A, an open session. So with that, let me introduce our first speaker, Bing Yang. I've had the pleasure of knowing Bing for probably about 15 years, since both of us were in our early days, respectively, at Iowa State. And Bing has been, very significant in the role of studying tail effector biology, including natural systems, natural pathosystems, rice and bacterial blight being the primary one, as well as engineering resistance mechanisms to bacterial blight through genome editing and other means. So he's now a professor at Mizzou with a dual appointment as well to the Danforth Center, and continuing to look at resistance in rice to bacterial blight, as well as genome editing systems. So I think we'll hear about both of those topics today. And Bing, thank you for being here.
I'm not as Aaron is. Good morning or good afternoon. And first of all, I would thank the organizer for invitation, and also thanks Aaron for introduction. And I want to talk about a couple of things. Like, one is the genome editing, particularly prime editing, which I think is very robust and powerful, and most of, especially student or postdoc, should be apply this technology in the research, in your research. And second, how to apply the technology for some real-world problem, or how to solve the problem. And go ahead with, we point out challenge, especially there are lots of challenge, but the grand challenge will be focused on one is the population increasing, and which means we need this population, including ourselves, need more food, and also the dietary composition change, need more of some special food. And the second, the land or farmable land shrinking, and also the extreme climate change, which all pose of some challenge to us. And what's the solution would be? And certainly will be create or recapture of some natural genetic variations, and can increase or maximum the genetic potential for the crop production, especially in agriculture, and also can engineer some of the tolerance to the biotic or abiotic stresses, especially the disease, which I think will be related to the work we have been working on. And as all of you know, I don't want to spend too much of time. I'll talk about the genome editing. This is kind of the early stage of the genome editing focused on the double-strand break and also repair of double-strand break through the intrinsic cellular repair mechanism. One is involves those specific endonuclease, which involves the site-specific recognition and also double-strand breakage. Those double breakage can be re-enjoined through non-homogeneous joining, which is error-prone, can cause insertion or deletion, which is the randomized and cannot be precise, and the second through template or homology-based recombination through this template. And all those could be involved the gene replacement. But there's, for the last several years, there's some new technology or new genome editing, especially based on the CRISPR-Cas, without involve the double-strand break and also involve without the DNA template, but the RNA template, that's the prime editing which. So the most popular or major one is the base editor one is like adenine-based editing through the dCas9 fused with the deaminase, change the nucleotide chemically, and through replication can make the base pair change from A to G. That's called the adenine base editor. Another one is the cytidine base editor, which is the cytidine deaminase fused with the dCas9, and then that's makes base pair change, and that change has a window of a few base pair related to or force within the guide RNA recognition site. So those could cause, those change could involve multiple sequence change in that space. At the same time, the efficiency is variable and has the context effect. For example, they almost focus on the C2T and also the off-target effect for the higher one, especially like a cytidine-based editor. The third one is called the prime editing. That's very particularly case involves the DNA Cas9 with the leakage Cas9 and also the RNA template to involve the guide RNA spacer sequence record specific site and also 3' extension for template, which could use for integration of the site-specific base change. And specifically for Cas9 or for the base editor, there's two major components. One is the effector protein, which is the leakase Cas9, and also the reverse transcriptase. The most popular one is the MLV virus because that's the most potent reverse transcriptase. And then the second part is the guide RNA, or called the prime editing guide RNA. It consists of the Cas9 scaffold, which could be recognized by Cas9, and at the 5' and at the 3' there's like template extension. That extension can consist of the two parts. One is the primary binding site, which can form the base complementary with the genomic strain, which from that R loop. And the second part is the reverse transcriptase template, which could contain the base pair change which we needed. That could be result the insertion, deletion, substitution, and all sorts of the conversion and the translation of the base change. So that's very precise or precise, but there are some limitations. This limitation could be larger, or how large the insertion could be for lock-in, how large could be deletion. So there are some limitations. So once those two components come together, recognize that guiding RNA is recognize the genomic site, and then they loop the double-strand break form R loop, and then the 3' end will be looped back and do the complementary with the genomic sequence, and then that genomic and then the reverse transcriptase could use that leaked 3' end as the primer to reverse transcribe the DNA sequence and form two flaps. One at the 3' flap, which incorporates the sequence change, and another is the 5' flap, which is a wild type. So this 3' flap and the 5' flaps have the equilibration, and sometimes if the 3' 3' flap incorporated, and you have the sequence change. And if the 5' prime flap and got in, and then you have the wild type. But if you have this continuous effector and guide RNA complex there, and then they continue cut and make the edit. But once they have the cut, and then the guide RNA sequence cannot recognize that site, cannot recognize the edit sequence anymore, and the edit fixed. So with that cycle, so that's why sometimes the efficiency increase or incrementally increase. There are multiple versions of the prime editing. The previous one, or the initial one called the PE2, is very inefficient, but through multiple labs, especially from labs around the world, and then they improve the efficiency. Now the latest version is called the PE7. So as you can imagine, there are multiple versions of the PE, and those PE contain different of the version of the improvement, and gradually, and now the efficiency is very high, which we'll talk about today, is the 3 version. And take one step back, when I work as the bacterial blight as a model to study microhost interaction, called the bacterial blight of rice, and it's the Pseudomonas pathogen, Pseudomonas oryzae, we call XLO, infect the rice. And this is the vascular disease, and it's a big problem for South Asia and Africa, and cause lots of yield loss. And also we use this as a model. I think this will be the best crop disease system have been ever done. And we particularly focus on, as Aaron to talk about it, like we particular focus on the tau effector is the group of the bacterial protein as it produce and secrete into the host plant rice, and then find the toxide. Most of the case will be susceptibility gene as the promoter of the rice, and then can cause disease. So this tau effector has the essential repeat, which consists of 34 to 36 amino acid identical or almost reserved repeats, and most repeats are identical except 2 amino acid at the 12 and 13 position. We call the repeat diverse amino acid. And those repeats, those 2 amino acids determine the specificity for DNA binding. That's the prototype of the tannin, which can be reverse engineered of the protein and based on the DNA sequence. And for this to use the tannin or later with the CRISPR to do some of the bacterial blight disease engineering, we need to understand the biology. So this is the model or working model for at the molecular level for the bacteria or XLO and the rice. When the bacteria secrete the tau effector and target the sweet gene, for in this case is the sweet gene, is the sucrose transporter gene, and once the gene activated and the plant becomes susceptible, and then this is the compatible. At the same time, from the right side, they have the promoter mutation, which prevents the tau effector binding to the sweet gene, and the plant becomes totally resistant, which is resisted as the dominant resistant gene. So that's the arms race between rice and the bacteria. At the same time, the bacterial tau effector can depend on the general transcription factor. One of the general transcription factor is the TF2A gamma subunit. In rice, there's one mutation, one base pair or one amino acid change converts the TF2 gamma subunit into the resistant gene XF5, which is the recessive susceptibility or resistant gene. At the same time, the tau effector could be trapped to target the defense gene or killing gene, or we call the executor resistant gene, can cause resistance. And then the third one is from plant have the typical nuclear binding site called the NLR. Type R gene can recognize the foreign tau effector, could trigger resistance, is represented by the XA1 allelic. At the same time, when the bacteria can to avoid this NLR recognition and can trigger resistance, can truncate the 3' or C terminal of the nuclear localization signal and activation domain, can avoid the recognition, at the same time can actively suppress the resistance, triggered by the fullness tau effect because there are multiple tau effects in bacteria. Some of them could trigger resistance from the NOR. So this is the arms race between those two components. And particularly today, I want to focus on two aspects of this interaction and based on the prime editing to do the resistance. So one is based on this XA1 or TF2A small subunit. There are differences between, it's just say only one amino acid or two nucleotides. So that provides the perfect example for prime editing because we just only need to find the two base pair change. So this is the gene structure, and then the XA5 contain one nucleotide change from the V to E, and the two nucleotide change. If that two nucleotide change create one base pair or one DNA recognition site. So we design the guide RNA and use the prime editing to make the guides and same time and make the construct to the transmission of the rice of Kitaki, which is a susceptible rice or contains the TF2A gamma subunit. And then with this genotyping, we PCR out the region and then digest with a restriction site, which enzyme called the SML1. With that restriction site digestion, which means there's edit. If there's no digestion, it's a wild type allele. So through this rough genotyping, we can see the efficiency is very high and could reach about 80%. 80%. So that's very high. As you can see here, there's 26 T0 plants. There's biogenic, about 31%, which means two allele of those two nucleotide changed. And there's monoallelic is about 50%. There are only 3 out of 26 are wild type. So there's one has undesired edit. So which means the efficiency is very high, and then we did the sequencing, and then the sequencing come out at exactly two nucleotide change or one amino acid change. So that's the case. And then the second strategy will be knock-in strategy, like I said. But the rice contain the R gene could trigger tau effect to activate the R gene, but there is some R gene or susceptible allele which does not contain the tau effect binding site. So if we can put this tau effect binding element into that susceptible or non-functional executor gene, then we can engineer the resistance, which is corresponding to major variance tau effector. So in this case is the XA23, which is a non-functional XA23, which does not contain the corresponding tau effector XA23, AVR XA23. So in that case, there's AVRX7, PTHX3, which are major tau effector and can target the sweet 11 or sweet 14 promoter element. So that contains 30 nucleotide. If we can put that 30 nucleotide in front of the non-functional XA23, and then the XA23 should be functional, once the bacteria infect the rice, the bacteria contains the AVRX7 or PTHX3. So that involves 30 nucleotide insertion or lock-in promoter of the XA23. And at the same time, that region or that 30 nucleotide contain two restriction sites. One is the XEXA1, another is the BLP1. So by PCR out the region from the T0 or from the rice plant, and then subject the PCR product for restriction digestion, we come up with a rough estimate of the edit or and as you can see, the efficiency is very high. And there's also about 70%, monoallelic about 55%, and then diallelic is 18%. So that's roughly 73%. And then even with the prime edit or with the edited side, and the precise is very high because sometimes we end up with the byproduct, which because the reverse transcription was going on and can create some of the can revise, transcribe some of the sequence from the guide RNA scaffold, and the scaffold could end into the site. So that's called the undesired edit. But the desired edit efficiency is very high, in this case, 47%. And the sequencing or deep sequencing of the results shows exactly 13 nucleotide insertion. And then the phenotype for both cases are expected and for resistant to certain of the bacterial strain which contain tau effector or certain bacterial strain which does not contain this tau effector or corresponding tau effector. So I don't want to spend good time to explain this. The message is the edited plants, once they have, especially for the XS5, which is the recessive, the homozygous or biallelic, especially in T0, could be resistant, and monoallelic or wild type are susceptible. At the same time, when XA20, non-functional XA23 has the insertion, which is the dominant and can be resistant in T0 with 1 or 2 edit events. And then the sequence, the next step is if the efficiency is so high for single, can we just push the envelope to multiplex of the guide RNA or multiple prime editing? So in this case, we made the modular assembling, which with individual guide RNA unit which can be used to assemble up to 4 guide RNA or peg guide RNA. And in this case, it can be assembled to 1, 2, 3, and up to 4. So to do that, and we designed pick up 2 sites, like the XA5 and XA23. Both sites could be for disease resistant with first test of the 2 guides. And we put 2 together, as you can see, the efficiency is very high in T0 or primary generation. As you can see, the monoallelic and diallelic is pretty high, and they have a combination of the single, and the double. And then the efficiency with 2 genes or 2 toxide got edited is like 46%, which is pretty high. And in this case, the sequence perfectly for as we expected. And the plant has the expected phenotype. And the last one will be like a quadruple or make the 4 guides, which is still those 2 guide RNA or 2 toxide for prime editing at same or for disease resistant, and another 2 for herbicide resistant. In this case, with the AOS and EPSPS, which could be resistant if edits will be resistant to herbicide. And this is the result of the biallelic, monoallelic, and the wild type. As you can see, the wild type this is like without all plants, every individual plants got edited in terms of different of the number, has like 1 guide RNA or 1 toxide got edited, and 4 plants with 2 guide or 2 side got edited, and 3 plants has like, or 8 plants has the 3, and 10 plants has all 4 edits. So in that case, the editing efficiency is very high, and with like 40% of the plants or T0 plants got all 4 edits in terms of the mono or biallelic edit. And the plants are resistant to bacteria, blight resistant. At the same time, the plants also resistant to herbicide resistant. And then this is the bacteria disease resistant. And then we did the whole genome sequencing of multiple plants. We sequenced the wild type plants, and then the plants contain wild type genotype, but through the tissue went through the tissue culture and transformation, and then the plant with the most efficient edits. And in that case, trying to figure out if there are any edit, like off-target effect of the whole genome sequencing, and analyze turn out to or reveal it's no specific off-target or guide RNA dependent toxicity, because there are always variations even through this tissue culture and transformation, but there's no association of the guide RNA and then the variation or the mutant. If you see, that's reasonable because this guide RNA make single strand DNA instead of double strand. Even there's like off-target or some mismatch or potential off-target, they create the single strain, and the single strain can be easily fixed by repair. So in that case, the off-target effect is reasonable with not associated with the guide RNA. And also we particularly analyze this potential has like 1 or 2 nucleotide mismatch. There's no off-target associated with the guide RNA. So with that, I would like stop here and acknowledge the graduate student A.J. Gupta, and he plays the instrumental role for this prime editing established and also application for this part, and also Bo did the transformation and the whole group, whole lab, and help with this, and funding from different source and collaborator from Qijing Chen, which from initially made that guidance or pig guidance or like PE 5 max system. With that, I thank you for your attention. Should we have a question, or we just...
Yeah, we have time for a couple of questions.
Oh, yes, another one.
I wonder whether you have anything in the field or the rice bacteria with this.
Have the...
Anything that promising, performing well in the field?
Well, so we have the rice genome editing plant or line based on the CRISPR-Cas9 by edit the promoter elements of the sweet gene. But for prime editing, we just demonstrated this feasibility with the Kitaki, which is not cultivated rice, and now we're currently doing the IR64, which is the mega cultivar, and which could be used for field test, but not in the field test stage yet. Thank you.
Thank you.
Bing, I have one question for you. Did you test, those efficiencies were very impressive. It seemed like it was just working. Did you test variants, or did you have to develop the efficiency to get it to that level?
So like indeed, the efficiency variant based on the target site, and because there are so many of the combinations to design the length of the 3' ends, or pick up the primer binding site, and also the length. So there are variations. So indeed, there are some variations, even some site which corresponding to the Cas9 recognition, which is very poor. And for example, now I think we did probably around 15 to 20 target sites, and there's a couple of them we never got hit.
So those are your best targets?
Yeah, those are those are published there. So as you know, for publication, you probably pick the best or sub-best one, right?
Thank you.
Yeah.
Introduce our next speaker.
Thank you, Bing Yang, for sharing your research with us. I would like to introduce our next speaker, Dr. Eric Kimmick, who is the Executive Director and Chief Science Officer at the Gene Editing Institute at Helen F. Graham Cancer Center and Christina Care. He is a pioneer in molecular medicine and gene editing fields. His research focuses on understanding the process of gene repair and gene editing with a focus with CRISPR-directed genome engineering in human cell lines. His team has helped create the first CRISPR gene editing tool to allow DNA repairs outside the human cell. Additionally, at the Gene Editing Institute, they collaborated with Delaware Technical Community College to develop the first ever gene editing curriculum for community college students in Delaware and across the nation. He has more than 155 publications and 18 patents to his name. Dr. Kimmick holds faculty positions at both the University of Delaware and at Winster Institute. He has been awarded numerous awards, including the Proudford Research Award in sickle cell disease and many other countless awards for his pioneering work in the gene editing field. Please give a warm welcome to Dr. Eric Kimmick.
I don't think that cuts into my time, right? The 20 minute, okay, thanks. I just want to thank Jasmine, Aaron, and the Society for inviting me. I'm going to take a bit of a right turn from some of the really interesting talks we've heard on gene editing and give you some perspective on being in this field for a long time and working in the field of human cells, particularly cancer cells, and how we think about gene editing and how it can be used. And then I hope show you some landmines to have you avoid stepping on going forward, because at the DNA level, regardless of the cell type, these things really exist. So just a little bit about what we are. We have two divisions in the Institute. Christianic Care is a 14,000 person health system. It's the hospital of the President Biden. He lives about 10 miles from where we are, and I think he's home today. We never know, because the whole state closes down when he comes home. And so we're divided into two groups. One is translation and discovery research, and the other one is educated and outreach. And one of the things we felt that was important is, as we develop these sort of clinically applicable gene editing directions, we certainly don't want to leave anyone else. So our education group actually builds trust and making sure that there's a diverse population awaiting these kind of very serious genetic advances. So I just want to spend 1 second and tell you kind of where we work. This is actually a famous quadrant from Don Stokes. It's called Pastor's Quadrant, and it defines science into 4 different blocks. The first is considered to be the quest for fundamental understanding, and Niels Bohr is a good example of that. Bohr was more interested in just pure basic research and understanding how things really work. And then in the lower right-hand quadrant is Thomas Edison, who people believe was more involved in pure applied research, even though we now know that's not exactly true. But his consideration of use was quite high. And I think all of us these days, particularly my laboratory, works in what's known as use-inspired basic research, which Louis Pasteur is an excellent example of, and he's the upper right corner. Now, you know I've left one quadrant blank, a low quest for understanding, a low consideration use, and you might have a colleague or two you might want to put into that one, and hopefully by the end of this talk it won't be me. So as Jasmine mentioned, I've been in the field a long time. We started thinking about CRISPR and gene editing and single-stranded templates for changing genes in about 2003. I think the first publications began around 2000, and ironically, this is in JCI, and we're just about the number of people who are in the field of gene editing. My colleagues were running after viruses to cure genetic diseases, some of which were pretty successful. So I think one of the things about gene editing that has evolved, and I get asked this question a lot, is just kind of the toolbox that we had back in those days and the toolboxes today have increased highly in precision. So we've gone from something that looks like a blunt instrument up to the Swiss Army knife. I think some of my competitors might believe I still work in the blunt instrument area. But I think now we have, and we heard today from Bing, some really great work in prime editing in plants, and so the tools we have now are actually really exciting, but they aren't perfect, and when you develop something going forward for trying to understand, especially with the treatment of a human disease, unfortunately, they emerge quite quickly. So just to level set everybody, and we've had many different discussions this morning in a really excellent gene editing session, and then Bing went through this very nicely. But we kind of think about gene editing as a genetic spell checker in which you can cut, paste, or change a word on your computer, and now we have the tools, believe it or not, to delete, insert, or replace, or knock in a specific gene or base itself. Now, to be fair, the only thing we can really do efficiently with CRISPR is knock a gene out. We think we can kind of repair genes, but I'm going to tell you a couple of secrets that have just emerged last month from the major CRISPR meeting in Copenhagen about inserting genes and even fixing genes. It's a little bit more complicated than we think. But remarkably, CRISPR, evolving from talons and zinc finger nucleases and meganucleases and every kind of other beast that was there many moons ago, actually is absolutely incredible for disabling genes. And we think we kind of understand the indel code now and how it actually does so things really efficiently. Again, as a number of people who preceded me during the day and the gene editing talks and today in this session, the world is divided into two parts, non-homologous end joining and something that's a really horrible term, we call this a basket term, homology-directed repair. That means we don't know how it works, so we just kind of throw in a bunch of reactions. That's really a little bit more on template DNA, and it's a little bit misleading because a lot of folks, especially in the mammalian world, actually use templates for gene knockout as well, and we do too. So my talk today is going to focus a little bit more on what happens with the break, how does the cell process the break, and what the break tells us about how the cell is trying to manage these CRISPR-induced breaks. And you can do these breaks and do these insertions plus or minus a template. So you can sort of pick your favorite direction. This is really just to bring us all together. We're all involved in this process, whether it's in medicine, biology, or biotechnology, and amazingly, this is probably the only meeting I've been at in a number of years when all of these the quality scientists who do this for a living are in the same room. So it's really a great opportunity to visit some systems that I don't really read about, but sort of some of the talks were really inspirational this morning. Well, here's what we study. The basic problem in cancer and in treating cancer is that I think if you ask surgical oncologists or oncologists, we actually have enough drugs. It's not, and the problem is not the drug. We have enough cell therapy. T cells are remarkable advanced antibodies. But the resistance to chemotherapeutic agents or just about every sort of agent we put in the body is rather quick. And I just put this one up here as the resistance to chemotherapeutic agents. And on the bottom is the date of the discovery, followed by the date of FDA approval, and then followed by the date of resistance, when the resistance to that drug was identified. And it doesn't take much hard looking to show that these are very close together. So the minute that a new drug comes on the market, especially chemotherapy, which is still used in a majority of cancer patients, you could put radiation therapy in there as well, resistance of the tumor cell develops. And this goes into something we call the therapeutic cycle. So, and I hope none of you have experienced this, and I'm afraid that some of you probably know people who have, that you get a tumor, you get diagnosed, you get a therapy, and it goes away for about a year. And then it inches its way back out. And the doctor said, that's okay, we'll increase the dosage, and now you have side effects, and you start to have radiation burns, you might have even chemotherapeutic effects on your body. And now you start to go, but in some cases it actually works, and you feel good again, and then it cycles back up, and on we go. So that problem is constant and has been with us really since cisplatin was introduced in 1971. So a drug that's used today was introduced in 1971 in chemotherapeutic suites. Kind of amazing. This is the culprit. This pathway, NRF2 KEAP1, is the pathway that causes these problems. And so for years, people tried to develop drugs against NRF2. There is a drug dedicated to KEAP1. KEAP1 is one of the targets of Keytruda. So Keytruda is a drug developed by Merck. It is now going off patent, so they're scrambling pretty quickly to find something else. So you used to see commercials with Keytruda solved all the problems. Now it's Keytruda and radiation, Keytruda and chemotherapy, Keytruda and immunotherapy, and that's because they're trying to find new ways to go. But it is a good drug. The partner for that target is NRF2, and what happens is KEAP1 keeps NRF2 in line because it's a bad protein. It's a protein that will cause resistance to just about every sort of cellular stress. So when you're being chased by a saber-toothed tiger in the ancient days, or you're trying to cross the street out here in front of the hotel, your stress goes way up. Well, that's NRF2. NRF2 is trying to protect you from doing something dumb, like disagree with your wife or your partner or something like that, right? And under normal conditions, that works fine. However, when KEAP1 gets mutated or the stress levels go up and ubiquitination of KEAP1 occurs, NRF2 is unleashed on tumor cells, and that's what blocks the chemotherapy from working. So that's why you have the cycle. It's a pretty simple system, as usual, developed by nature. Here's the problem. NRF2 suddenly starts to activate oncogenesis in a lot of different ways. We studied drug resistance, and we just drifted into invasion of metastases in glioblastoma. But it also induces inflammation, proliferation, angiogenesis, and just about everything you don't want in a protein to be doing while the doctor and the oncologist is trying to treat you. So the counterbalance here is really pretty remarkable. So we've been working our way through the FDA with a simple idea that we'll develop a CRISPR-Cas product, CRISPR-Cas molecule, that will specifically disable NRF2 so that we would then be able to continue treatment with chemotherapy, radiation, or targeted therapy. And actually, it's gone well. We were pretty surprised. We're about halfway through the approval process. I never want to do it again, and I'm told it's going well, so you can imagine when things don't go well. But one thing that the FDA asked us to do was to consider the target, and this is actually a really key part, and that is, what is the indication that's really going to help people? So we started with a very small population of lung cancer patients. We've moved into head and neck and esophageal cancer, and that's kind of made us think about how best to use the CRISPR drug, and that decision actually led us to some interesting discoveries because we moved targets inside the NRF2 gene. Here's how it all falls out. There's a discovery, preclinical, IND-enabling, and clinical phases you go through. We're through the Interact meeting, and we're right at the edge of the IND-enabling studies, and that's where you're engaging manufacturers and people like that. But the reason I put this slide in is it's constantly reviewing your data and your best targets. And one of the targets that we thought about using right away actually isn't the target we're taking forward. And so when we made that change, I want to show you what we discovered. So from here on out, I'm just going to tell you some of the landmines that can exist in this thing. So does it matter where you edit the gene? It does. Here's the gene. Here's NRF2. It has 5 remarkable exons. The first exon is so small, people generally have not targeted, and we've done most of the work on NRF2 targeting, so we've probably developed over 200 to 300 guide RNAs for just about every sort of Cas protein. And we always come back to strep pyogenes Cas9, believe it or not. It seems to be the workhorse of all of this. So exon 2, 3, and 4, and 5 are targets that we've done. So decide the gyne, transfect the cells. We're actually using a lipid nanoparticle to transfect the cells. And in the first round of targeting, these are pancreatic cancer cells. And I just want you to look at the green and blue. So exon 2, targeting is very good, but slightly different. The different colors indicate the type of mutation that it makes, exon 2 and 3. Here's exon 4. Okay, 2 different sites within exon 4. Look at the green. That's the wild type. So that's what's left behind. So the efficiency of targeting as you move up and down the exons is quite dramatically different. Okay? Now, here's a really important thing, and I know I don't know how hard this is on clonal cells in other organisms, but when you target a gene and you look at the population of the cells created, you may get a phenotypic response, you may get a really nice Western, you may do some cDNA cloning. The problem is you don't have one population, you have a lot of populations. And so what we spent doing was cloning at about 200 to 300 cell lines and started to look at the kind of macerations that go on. And here's a very good example. There are some wild type lines. Nothing has happened, right? The wild type is preserved because you don't get 100%. There are cells that have a single copy knocked out at -1 and the other one is a wild type. There's one with wild type, with +1, -4. There's all kinds of different heterogeneous populations. There are clonal populations inside the whole population that you're targeting, and that becomes a problem. Because now you're not dealing with one population, especially in the world of tumor biology, right? You may get a really nice killing, but now we know there are different populations in there, and they could be responding differently to your treatment. So as many times as you knock a gene out, something is left behind, unfortunately, and it's not a stem cell. It's a somatic cell that has a various population. All right, so these are lung cancer cells, which we have knocked out. This work took almost a year to do. And just look at the mRNA in the last column, right? That's a clone, and it has 3 copies of the gene. This is a NRF2, has 3 copies in this adenocarcinoma line. So we get a -2, a -3, and a -2, right? So they all look sort of the same. And then suddenly we notice that in some of the cases, exons 2 and 3 are missing. They're gone. So unless you do sequencing about 5,000 bases up and down, you are not picking up what you've actually done. That's the first step. Look it down in exon 4. 3, minus 1, minus 103, minus 103. Last one, the C33 clone, minus 101, exon 4 is missing. This is a process known as exon skipping, and it's common in every form of DNA we've looked at. So unless you're looking on different sides of your exons and in introns, you may be losing pieces of your DNA, and you're probably getting fooled. What we've decided now is to give you sort of a diagram. Even within the same population of tumor cells, and it is likely true in stem cells, we looked at iPSCs as well, and it sort of gives you the same thing. Occasionally you'll get the full NRF2. Then you'll get a population of cells that have exons 2 and 3 missing, exon 2 alone, exon 4, and then a shortened version that comes mysteriously out of North Dakota. We don't know where it comes from, and we can't actually figure out how that happens, but it's clearly there. So does genotype change predict phenotype response? So I want, when I target a population of cells, I want to be sure that that clever design, which I've done on a computer, and maybe even using AI, will actually give me the rollout that I want at the phenotypic level. Well, maybe. Here's what happens. This is a little complicated, but let me walk you through it. On the bottom, if you see the indel, that will tell you what the 3 genes are actually like. Like the first group of blue-colored bars are all wild type. And then one with -2, -2, -2, -1, -1, and on, okay? Different combinations, and we call that the genotype, or the allelic content. And then the bars indicate the sensitivity of increased concentration to our friend cisplatin. That's your population. That's what's really going on in gene editing. Not a population where you just look at increasing concentrations, but when you break it down, a lot of things happen. And for our world, this is not good news. Okay? So we have to find a way in which the fourth one over, where it's the delta 4 minus 101, that's where we really want to. That's the kind of population we're going to get. So in the mammalian world of gene editing for cancer now, the press is on how do we develop what's known as the indel code. Here's another good example of it in esophageal cancer. This, I think, was shocking to me. SG3 targets exon 2 in NRF2. Its knockout efficiency is roughly 86%. Everybody's really excited about that, right? So SG76 and 83 target exon 4. Very large numbers, 89% and I think 76%. The response to chemotherapy is vastly different. Even though we have knocked out in the population, and you've looked at the genotypic level, or you've used Cas-off finder to look at off-target sites, you are not creating a stable reaction at the phenotypic level. So exon 2 deletion is the middle bar. It says 15 micromolar. It's the yellow bar. It has no difference than if you didn't target the cells. Something inside that population, there's a clone in there that is taken over that allows the cell population to avoid your treatment. Here it is in real life. So exon skipping, this is cDNA of the, and you can see in esophageal cancer cells, we target SG3, we get 2 bands. So there's been an exon looped out, and it makes a functional protein. If we move over to a FADU line, which is an oropharyngeal mouth cavity, oral cavity tumor, there's barely any editing in SG3, even though it's the same site. So even within different populations and tumor cells that are, legitimately less than 3 to 4 inches apart, the efficiency of molecular gymnastics to go on at this level is a serious landmine, and it's probably not unique to that sort of system. So here's the message. Kelly Banis, who leads this team and released in Copenhagen in April, she released something called the indel code. So what we believe is there are exonic enhancer elements up and down all DNAs, plant, insect, dinosaur, mammoth, human, etc. And each of them have little sequences that if you break apart, by no work of you can design these by AI, but if the reclosing, whether a template or not, of the break site puts these 2 bases together, you will skip your exon. You will induce exon skipping. And so you can either end up with an unedited alleles, you can end up with altered function or non-functional. All of those are contained within the same population. So, in conclusion, landslides and landmines. Aaron was asking me, what the hell is that title about, right? So the landslide is that there's a lot of information out there. Most of it is probably weak at best, okay? Pretty easy to knock a gene out. I'm a big fan of the rock group, the Eagles. And Joe Walsh, a guitarist, was asked, is it hard to play Hotel California? And he said, no, it's not hard to play, it's hard to play it right. Same thing in gene editing. It's pretty easy to do. You can order IDT or your favorite place and it'll work great. But be careful, because the population that's created, can cause all kinds of problems, and that's sort of your landslide. Understand and address the impact of on-site mutagenesis. Off-site mutagenesis is very complicated. Base editing, prime editing cause breaks in the DNA. Tony Kathman announced that in Copenhagen in April. Very disappointing to those in the human world that are trying to use base editing and prime editing without, that was not correct initially. We now know that it causes plenty of breaks in the DNA, almost as much as Cas9 does. And then the indel code. Intentional CRISPR design leads to unpredictable outcomes. We can't predict the indel. That means we can't control what happens, what the footprint behind is left by CRISPR cutting, but we can predict the outcome by knowing what that looks like. Exon skipping is very prevalent in targeted genes. I'm happy to talk to anybody who may be seeing that in your own targeting and other systems. I don't know if that's the case. And every target is clearly not created equally. So this is the team that developed the indel code and discovered exon skipping. Kelly Banas is second from the right in the front row, and Natalia Rivera-Torres is the group leader of the group. There's a guy in the back there with a gray beard. I don't know who that is. He might have just been walking by and decided to jump into the picture. So it's kind of weird. And then, amazingly, he stayed for the next one. This is our entire group, and I want to thank the funding agencies and all of you for listening today. Thank you.
Are there any questions at all? We have time for about one or two quick now.
Apologize to begin with. I'm a plant person, so this is going to be, it might be a little bit pathetic. This NRF2 gene, is it normally alternative spliced in any way?
No, that's whether that's from a plant person or any person. It's a great question. It's not. So you're actually inducing an alternatively spliced event by the breakage, and that actually occurs, I think, at off-target sites too. I didn't want to go into it today and take up any more time, but our off-targets for this gene are about 275. There is only one, so you have to go through levels of you have to go through a Cas-off finder and then a validation phase and then an experimental phase. And we've come down to one site, which is in one exon, but it itself is not spliced, but so far, until somebody invents a new technique, we have to do next week again, so. That's something.
Would you recommend screening for this exon skipping in a routine pipeline?
Well, we're trying to develop that because this happened a little while ago. So Carl June is a really great T-cell immunologist at Penn, and he was working his way through, and then one of our colleagues in Israel said, oh, by the way, Carl, the process you're using for making your T-cells, which are clinically approved going into humans, are invoking a translocation. So the trial stopped. T-Immunity, his company, laid off about 200 people the next day. So long way to say we don't know. Exon skipping can almost be something you have to sort of do the old-fashioned way by getting the cDNAs out, sequencing them, and looking at these gels. So these gels are just kind of, the general thing, but we sequence them through. Unfortunately, it's pretty common. The exonic splice enhancers are not a consensus sequence. They're little bits of sequences that we can't figure, at least we're not smart enough to figure it out yet. That's probably more apt. So unfortunately, I think the cDNA route from the mRNAs to see if you're getting losing sequencing. The only good news for us is that in knocking NRF2 out to enhance immunotherapy or cell therapy to work a little better, the exon skipping actually helps a little bit, even though it seems like it doesn't respond as well in a whole population, which is what a patient would see. It seems to be okay, but we're worried. Well, I'd be honest with you. It's been a surprise that that's been so prevalent, particularly in that gene, but it happens in KRAS and NRAS too. So.
All right, please join me in thanking Eric for the great talk.
I would now like to introduce our final speaker of the day, Dr. Aaron Hummel, who is the Vice President of Research and Development for Pairwise, and he's been working to improve crops through genetics for 15 years now. Dr. Hummel is a leader and expert in plant molecular genetics and genome editing technology. Dr. Hummel received his bachelor's in biology from Hillside College and his PhD in molecular, cellular, and developmental biology from ISU, or Iowa State University. Dr. Hummel did his postdoctoral training at the University of Minnesota. Dr. Hummel holds over 25 patents and patent applications and is a decorated veteran of the U.S. Marine Corps. Please give a warm welcome to Dr. Aaron Hummel.
All right, so we've heard some great talks about the application of prime editors in rice, considerations for gene editing technologies in therapy, and I'm going to try to wrap up the session with a talk that is mostly technology development focused, and I want to share some of the lessons that Pairwise has developed over the last 6 years in using multiple different kingdoms, very disparate species, to develop these editing technologies. So hopefully this will be a good conclusion to the session. So the outline, I'm going to give you a few brief comments on why Pairwise exists. One slide on why templating editing matters. That will not be a comprehensive assessment, but it's one of my thoughts. And then a story on the invention and optimization of our templated editing system, and some lessons learned from that and other work that we've done in tool development. And then one story about what I think is part of the future of genetic improvement in agriculture. So Pairwise is a technology-focused food and agriculture company. We mostly use gene editing to affect plant genetics in ways that create novel traits that benefit consumers as well as growers and supply chain participants. And the exact strategies and trait that we seek to develop depend on whether it's a row crop or a consumer crop and the specific circumstances of the plant that we're working in. We're located in downtown Durham. That's where our headquarters is. And we also have additional lab space in a greenhouse in the Research Triangle, which is nearby. A couple of significant points in our history. The first is that we've raised $115 million in Series A and Series B rounds. And we've had two exceptional collaborations with our partners, Bayer Crop Sciences. The first was, it started in 2018, and it was a 5-year collaboration that focused on trait development in corn, soy, cotton, canola, and wheat. And it was very successful. We created 27 novel traits that are now part of Bayer's development pipeline. And some of the successes there are increases in corn yield and also Asian soy rust resistance. And so the first collaboration was so successful that when it ended last year, we inked a new deal with Bayer that is also 5 years and focused entirely on developing short stature corn via gene editing. So just to try to give you a brief comment on one of the reasons that templated editing matters in eukaryotic systems. This table is a meta-analysis that compiles a bunch of different studies, and these particular studies are in common crop species You can see in the second column the number of individuals and some notes on the diversity of genomes that are selected in these studies. This is focused on crops, but you'd get the same or a similar answer if you looked at genetic variation in animals and in humans. And the main point is that the single nucleotide polymorphism is by far the most dominant form of genetic variation when you're comparing individuals in diverse populations. And it substantially outnumbers indels as well as structural variants. So the reason that matters to us is that we've found that natural genetic variation is by far the most valuable source of knowledge when we're trying to design alleles for gene editing. And so because of that, the abundance of SNPs in nature is significant to their importance in plant traits. And obviously, single nucleotide polymorphisms and even multiple nucleotide polymorphisms require the ability to precisely change those bases. So you can access some of that category with base editors, but to really fully access that full category, you need templated editing. So that's not a complete answer, but that's one of the reasons I think templated editing matters a lot. So 6 years ago when Pairwise started, we knew that we had very ambitious goals to do technology development and also that we were in a competitive field and that we were aspiring to work in plants, which can be kind of slow as far as test systems, as many of you probably know. So we deliberately set up a small human cell lab, also the capability to do direct measurement on enzyme biochemistry in a test tube, and a couple of directed evolution pipelines in bacteria and yeast. And we've used those three approaches together with transient and stable assays in plants to develop our CRISPR editing technologies, and using them in a coordinated way, we've been fairly successful in that. So one of the things that we managed to develop with that approach is what we call redraw, which is a novel templated editing system that enables us to make all types of small edits, including any base change, any multi-base change, small insertions, as well as small deletions. And I'll admit that redraw is at best a mediocre acronym. I need to actually compare notes with Mark Settles and see how the Pairwise acronym development compares to NASA, because I think they might be similar. But that's the name that we chose, and that's what we're going with. It does sound kind of nice when you don't think of it as an acronym. So the way it works is that it uses a CRISPR enzyme to make a double-strand break, and there's an extension on the guide RNA that actually reaches across the double-strand break and pairs with a complementary sequence on the target strand of the genome. And then the reverse transcriptase will come in and directly copy the sequence of that extended RNA into your target site, and you end up with an installation of your precise edit. So if you're comparing this to the talk that Bing just gave on Prime, you'll see that there's some obvious similarities, but there's also some very important differences. So just to highlight those, first, we're making a double-strand break, and second, and probably most importantly, the priming reaction occurs on the target strand away from the enzyme complex that's bound to its target site. And we've actually tested this with Cas9, Cas12a, Cas12b, and the proprietary enzyme Shark that Pairwise has developed, and it works with all of those systems. So every CRISPR system we've tested it with, it works with. And we believe that the architecture of getting that priming reaction away from the structure of the CRISPR enzyme on its target is why this system can work with type 5 and type 2 enzymes, and Prime seems to be limited to Cas9 systems. So on the right side, I'm showing the very first experiment that we ever did that actually gave us back positive data saying that this concept works. So this was in late 2020 is when we did this experiment. The leftmost treatment is just a negative control, and all the rest of them are variants of redraw. And you can see that the efficiency ranges from 0.2% to 1.7%. So not real impressive there, but as a first experiment, it was very exciting because it was a proof of the concept and gave us a design to focus on for further optimizations. We've put quite a bit of work into it since then. A couple years ago, we released a bioarchives preprint. If you're interested, that QR code in the right will take you there. And that kind of explains the early development efforts that we did on this system. Just to give you a couple highlights, we've shown that it can be, in human cells, it can make all 3 base substitutions. When you start with a certain codon, you can change it to any other codon sequence or motif. And that's obviously important because you can start thinking about doing saturating mutagenesis at amino acid positions for trait optimization. Or if you're doing disease biology, you could really explore that as well. We also compared it to homologous recombination using oligonucleotide-directed repair, and in the second generation of redraw, which is now kind of an outdated generation, we were getting 10-fold better activity for precise edit installation compared to the homology-directed repair, and also a much better edit to indel ratio. So since we released that paper, we've continued to work on it, and what I'm going to share here is some of the success that we've had with our yeast-directed evolution system. So what we were doing in this experiment is looking for improvements in the reverse transcriptase domain of the complex, and trying to find a faster reverse transcriptase that would work better for installing the precise redraw edit. And so on the left side shows the general workflow. You create a library that has genetic diversity in the reverse transcriptase domain. You introduce that to a yeast strain that has a mutated gene that requires amino acid supplementation to grow, and you can expose that to a dropout media. And basically what you're doing is you're requiring precise repair of that mutated gene from the redraw complex in order to get survival. So you're comparing the library at the end to the one that you started with, and you're looking for redraw variants that are enriched in the selection conditions. And we have 3 sources of diversity for our libraries. We can do the old school error-prone PCR approach, and then 2 different sort of strategies for designed libraries doing single site saturation mutagenesis or combinatorial mutagenesis with synthetic sequences. So this is how the selection system works. It's the ADE2 gene, and we replaced a glycine codon with a stop codon, which creates the strain that is unable to grow unless it has adenine supplementation, and redraw correction of that stop codon back to the glycine residue will enable the yeast to grow in the dropout media. And this is a representative snapshot of the type of amino acid diversity that we find after we do the dropout selection process. So the top bar is the starting reverse transcriptase, and each of those little marks is an amino acid mutation compared to the original sequence. So you can see that we're getting a lot of diversity there. In this particular example, we were selecting candidates based on having at least 10 reads in the starting library and an enrichment factor of at least 5, so 5 times more abundant in the ending library than they were at the beginning. And so we got a handful of candidates from that process, and then we took them into HEC293 T cells to test their activity in an environment that's closer to our application purposes. And so what we're comparing here is we have 2 different target sites at the DMNT1 site or locus in the human genome. The solid color shows site 1 and the stripes show site 2. We're comparing them to the dashed horizontal lines. The top one shows the activity of the starting reverse transcriptase at the site 1, and the bottom one is at site 2. And what you can see is that most of over half of the mutated reverse transcriptases are actually superior to the starting sequence. And a lot of them are in the 3 to 5-fold better range for activity than the starting sequence. Another interesting thing is that the majority of candidates that turned out to be better have only 1 amino acid mutation versus the 2 amino acid mutations didn't work at all, and 3 and 4 were kind of intermediate. And then also notice that we're now up to about 25% max editing efficiency. When we first made the redraw discovery, I showed you our max editing efficiency was at 1.7%. So ongoing and systematic optimization of these tools with these kind of pipelines can be quite effective in driving a higher performing tool. So some lessons on what I just shared with you, as well as a lot of the other tool development work that Pairwise has done. I guess the first thing is that if you have the capability in your organization to set up a coordinated workflow that involves multiple systems such as yeast, bacteria, mammalian cells, and potentially plants, if you can coordinate that into a pipeline, you have some significant advantages. For one thing, you can tailor your system to the particular characteristics of the tool that you are engineering in any given experiment. So you can imagine sometimes you want really high throughput approaches, sometimes you want high sensitivity assays that maybe you're using for rational design inputs, and you really want to be able to know what is absolutely better than the other variants. And all kinds of other things as well. And when you have the capability to use all these different systems, you can take advantage of their benefits. The next one is probably super obvious, that higher throughput in some of these systems, such as yeast or human cells, leads to faster success in hitting your performance milestones, just because you can test more variation in each cycle. And then more comprehensive data will enable you to get better intellectual property claims, if that matters, and possibly better publications. That's a hypothesis I hope to test later with the data that I just showed you and others. And so then some thoughts on why this works when you approach it like this. The first thing, I think Eric mentioned this as well, is that once you're inside the eukaryotic nucleus, the fundamentals of CRISPR, protein, DNA, and nucleic acid interactions seem to be highly conserved. And that's very well supported as well by the fact that over the last decade, the scientific community has been quite successful in spreading CRISPR tools all over the biological world and getting editing results in those systems. And the second point is actually kind of the opposite of that you really need to tailor and customize your delivery modality to the system you're working in, or else you're going to get poor results because you'll have poor delivery. And so the extreme example is probably using tDNA via agrobacterium in plants. You got to have a really well-optimized expression cassette to get that tool expressed in your plant. Versus therapy, you're likely using ribonucleoprotein complexes and delivering those via nucleofection or maybe even in vivo. Those are very different delivery modalities that require their own optimizations. And one thing that I don't want to go too deeply into, but I'll just mention here, is that Pairwise has experienced some differences in sensitivities to some tools. So base editors is probably our best example. The human cells that we've used have been pretty much impervious to the effects of base editors, whereas in monocots we've seen a little bit of sort of cytotoxicity of the base editors in the system, and they inhibit regeneration to some extent. And in dicots, it's even greater sensitivity to that. And we found sort of practical workarounds to that, so we didn't delve too deep into the biology, but it was an interesting observation that we found that doesn't seem to be true of cutters or other types of CRISPR tools. So last thing I want to share with you is a little story that I think points towards the future of genetic improvement via gene editing in crops. And this is a story that we actually did with base editors, but after I finish it, you'll see why it's probably going to be even more important in the age of templated editing as we get into the next few years. So this is from our first collaboration with Bayer. The intent here was to develop larger ears of corn that have more rows of kernels around them. And you can imagine if you get that scenario, you could have a plant with a higher yield potential than what you started with. So there's a gene called FEA2 that controls the inflorescence meristem of developing corn ears, and it controls the size of the meristem as well as the patterning that it lays down as the cells differentiate. And there were a number of mutations known in FEA2 that were early truncations, pretty much, or mostly frame shifts. And they all resulted in what you see in figure B, which is a fasciated ear that has really large number of kernel rows, but a severe shortening and complete loss of patterning, which is not a useful phenotype commercially. And there was one allele that was a proline to leucine substitution that was known that gave a moderate increase in kernel row number. But it was one of those circumstances where we knew that this had arisen in breeding, but we didn't know if that was the best allele or if that's just the one that chance had delivered to us. So we used our base editing toolbox to create a series of amino acid substitutions at that same position. And you can see that there in detail. And then we also used our cutting tools to create a small series of in-frame deletions at the same location. So this led to what we refer to as an allelic series. And then we tested the allelic series in the greenhouse, and the inbred genotype that we had been using, the unedited inbred genotype gives 16 kernel rows typically. And we found that each of our edited alleles gave an increase in kernel row number from 18 up to 24 rows. And so two things were interesting here. One is that we had translated the allelic series into a phenotypic series, and also the second one is that each of the alleles was giving a different number of kernel rows. So super attractive in the greenhouse, very exciting. We then took that, a subset of those alleles out to the field and found that a serine substitution was actually the best balanced allele that we had in this experiment, because it gave almost a 10% increase in kernel row count with no loss in ear length or 1,000 kernel weight, whereas the alleles that gave a more extreme increase in kernel row count also gave a loss of ear length and in 1,000 kernel weight. So, you know, I showed you this series of alleles that we were able to create with base editors. You can see on that detailed panel on the right that we were even pushing these tools to the point where they were giving us non-canonical base edits, and that gave us a bit more amino acid diversity than we should have expected to get. This is a highly optimized base editing toolbox, and we pushed it to the absolute limit, and this is all the amino acid variation we could get. You can imagine with a templated editing tool doing a complete saturation at this site and really having, you know, 20 different alleles, amino acid substitutions with which you could optimize the quantitative trait phenotype, and that could be a really exciting future for trait optimization in plants. So the last thing I'll say is that all of the tools I just mentioned and a bunch more are available for out-licensing from Pairwise, and we're also interested in collaborating actively with different organizations to develop traits for your pipeline. So if either of those sound interesting to you, please either send me an email at that address or come talk to me afterwards. I'd be happy to chat. And the other thing is that we've recently made a lot of progress in BlackBerry, and Deepika is going to be presenting an interactive poster tomorrow at 1:30 p.m. Sharing some of the progress that we've made in BlackBerry. So please be sure to check that out as well. So appreciate your attention. Hope that was a good closure to the session, and I think we have a couple minutes for questions.
Do we have any questions at all? And also, you can ask questions to the two other speakers too, because we have about 10 minutes left of the session if there's any questions at all.
Thanks. Thinking about me here, right here.
Yeah, hi.
Thinking about cross-kingdom applications. I'm assuming that you're codon optimizing for each application. Do you test out different codon optimizations each time, or do you just kind of roll the dice and go with one?
Yeah, when it matters, we definitely do. Like when we're at the endpoint to use it in plants, we test several optimizations and find the best one. In human cells, we typically don't test a lot because that's not our end use unless it matters to the experiment.
And just a quick follow-up. When you are testing those different codon-optimized versions of your base pair changer, or editor rather, is there a go-to kind of phenotype or experiment or edit that you use as a pass-fail, or does that depend on your application?
Yeah, we just go to the endpoint and we look at, we have some favorite targets that we're very familiar with, and so we just look at the editing rate at those targets and kind of the endpoint assay. Yeah, hey, Mark.
Yeah, hey. So for your last story on FEA2, that allele is a recessive allele, is that right?
Yes.
So,
well,
so you haven't done hybrid trials yet?
So, ish. It's complicated. I can't share everything I know about it, but it did turn out that in some circumstances it had semi-dominance. And it wasn't necessarily predictable. So we were expecting it to be fully recessive, and it didn't turn out quite that simple.
Yeah, so, but you didn't do any hybrid trials, is that right?
We have done hybrid trials.
You have done?
I didn't show them, but we have done them.
Okay, does it give the same effect in terms of increased kernel row number? Does it have an actual positive effect on yield?
So it's still in Bayer's evaluation pipeline, and I can't really tell you more than that.
Are there any other questions at all? That concludes the joint session. Thank you guys all for coming, and thank you again to all of our speakers.